Andrew Huberman has become a good friend, and it's interesting because he swims with sharks, creating virtual reality experiences for people to desensitize them from being fearful, and is an expert on many things optic and eye-related. I think of him as just like a nice, a guy who I get to hang out with a lot these days. And he's been helping us a lot recruit people for a BrainMind over the last year, but amongst other things, you've heard about brain-machine interfaces now, from Mary-Lou Jepsen using infrared light, lasing it essentially through a hologram. You've heard about Ed's use of sound and other things, or using optogenetics and things like that. Those are obviously not consumer-friendly ways to hack your mind, because you need equipment for them, at least for now. Another way to hack your system is simply controlling your breathing in new ways. And some of these techniques are old, some of them are new, and Andrew is an expert at many of them, and will teach us one actually here right now.
Okay, well, thanks so much to the organizers for giving me the opportunity to speak. I'm going to cover some data that gives some impression of how I arrived at the sorts of tools that I'll share with you in a kind of actionable talk here. I find myself in the somewhat uncomfortable position lately of both wanting to do very rigorous neuroscience, which I believe my lab is doing and will continue to do, but also in front of people in the wellness community and military communities, and interacting with them. And you learn that neither community has it exactly right, because as we've heard many times today, that we need very rigorous, detailed protocols before we can make things actionable. And yet there are a lot of people out there doing interesting things that may have kernels of value and knowledge in them that we could perhaps use and extract in the laboratory to develop even better protocols. So if that was all very cryptic, I'll just begin, and hopefully it will become less cryptic. So my lab works on these three things: visual development, how you wire up the visual system; visual repair, how you fix it in blind people, in particular people who are suffering from glaucoma. We run clinical trials. We also do animal studies. And then today I'm going to talk about visual processing in internal states, that is, how what you see interfaces with how you feel to determine what you do. And the reason why I'm focusing primarily on vision is not just historical. It's also because the number one determinant for what you do is what you see. As humans, we're extremely visual animals. So let's say we were to go take a walk up at the dish after this. We'd see an animal perhaps similar to this. At this point, our behavior changes, right? So our behavior is unconstrained, and all of a sudden it's constrained. The logical adaptive choice is to pause and maintain awareness, right? You wouldn't want to run. You could draw attention to yourself. You want to limit your detectability. Okay, there's a single and clear and adaptive response. You could also duck down and hide, but then you no longer can monitor the threat, okay? Assuming you view this as a threat, which I do. The game has now changed. The visual perception has changed only slightly, but through a pseudo-empathy, theory of mind thing, whatever you want to call it, you see this animal's eyes and you assume it sees you. You can stand there all day and hope it moves on, but perhaps your behavior is now more adaptive if you were to leave and hide or seek shelter. The game's changed again. So you could pause or you could hide, but probably the best thing to do would be to run or walk backward very slowly. And at this stage, pausing, hiding, or running are not good options, and your best option is to truly fight for your life. Okay? So the reason this is interesting to me is because the visual perception in the context varies dramatically between them on the one hand, but the internal state, you could imagine, might also vary. And a lot of what's been studied in visual neuroscience is about how we detect stimuli and make sense of them. And we realized in wanting to think about how we analyze visual threats that we were probably going to have to start thinking about organs outside of the brain, and that's the interface with both the brain and organs in the body, like the lungs and heart, and as they relate to arousal states, or some people like to call it stress. So I'm going to switch back rather flexibly. Forgive me if that doesn't fit exactly with other people's notions of arousal and stress, but for sake of this talk, it should work. Okay, so if we really want to understand how we go from what we see to how we, and combine that with how we feel to determine what we do, we need to consider the body and the brain and how they're working in concert. Okay, so perhaps the lungs and heart, how that's all integrated. And so we do this first in the laboratory in mice. So I'll tell a quick story that in the world of a mouse, this is a bad situation. And the mouse needs to make very good decisions about things overhead because lots of things fly overhead. There are trees overhead. There are many things that aren't threats, and animals need to forage. And believe it or not, mice do spend time out in the daylight. And so you can recreate a mouse's natural innate response to an overhead so-called looming threat in the laboratory the following way. So this was a protocol developed by my postdoc when she was in Marcus Meister's lab. So here's a mouse hanging out. A loom comes on, it pauses, it runs in, and then very quickly it rattles its tail against the wall. I'll return to that tail rattling behavior. Mice do this without any learning. They do it the first time, they do it every time, at least for a few days, until they realize that it's not really that threatening. This is the other response. The mouse is stone cold frozen. You'd almost think I'd pause the video, except that the loom is continuing. So mice either freeze or they run and hide. And that's the natural response of the mouse. So we were very curious about where the detection of the stimulus and the internal state of the animal somehow get put together. So we decided to screen the brain for areas outside the visual system and outside the traditional arousal system that might put this sort of information together and lead to intelligent, let's call them adaptive behaviors on the part of the mouse. So we exposed a lot of mice to these virtual threats. Then we screened the brain for genes that are activated in neurons that were recently active, so-called CFOS for the aficionados. And we found this nucleus or collection of nuclei, nucleus reunions, in something called xiphoid nucleus. And even though I teach neuroanatomy, I'd only heard about these once or twice. There's not a whole lot of information about what they actually do. Turns out these are just reflex neural recordings. If you record from this structure, it becomes more active under conditions of visual threat and other types of threat, auditory threat, olfactory threat, et cetera. So my graduate student, Lindsay Slay, mapped all the inputs, and the short story is this structure gets input both from the sensory areas that detect things and from the areas of your brainstem that are called, well, let's call them arousal areas that lead to sort of alertness or sleepiness depending on their activity. It's a very interesting nucleus, not a traditional visual nucleus. So then she went ahead and she activated this structure, and what she found was the following. This mouse is so-called ventral medial thalamus, that region that includes those nuclei, has been activated such that its activity is way higher than it normally would. And instead of running away from this threat, the animal is tail rattling, which is actually in mice a sign of threatening the actual the predator. Or you see this when you put two mice together and are about to fight. So instead of running and hiding or freezing, this mouse is now stepping out into the open and remaining in the open and saying, okay, let's go, let's fight. And we know people like this, right? And there's a healthy context to do this, and there's an unhealthy context to do this. So silencing the structure didn't lead to tremendous changes, but activating it in the following manner did. So we thought that was extremely interesting. I'm going to give you the short story here. Lindsay, I'll skip the quantification, but what she essentially saw is not only do mice now go out and tail rattle quite a bit more than they did before, but they also do that out in the open. They're not afraid to stay out in the open, which normally mice don't do. You can think of different types of confrontational threat, historical ones like Rosa Parks or the scene from Tiananmen Square. The confrontation of a threat that could somehow hurt you or kill you or change your life in a negative way is something that can have, depending on how you view it and the consequences, it could be adaptive or non-adaptive. We could discuss that offline. So Lindsay went on to map the outputs of the different structures here, and she found something very interesting, which is this red pathway that I'm showing here is the freeze pathway. So you have a pathway in your brain that takes what's going on and integrates it with how you feel inside. I'll talk about that in a moment, and makes you freeze or pause. It puts you into paralysis, which might actually be the healthy response. If somebody walked by that window right now with a machine gun, which is unusual for this campus, the intelligent thing for me to do is probably pause and see if they continue, not to hide, because if they come in here, I'm more vulnerable. The other structure in the output pathway is the green pathway, and that caused confrontation. And so we asked the question, what is this pathway doing? And I'll simply summarize by saying it's shifting the state of the animal. And we know that because if you trigger activation of this pathway, this confrontation pathway, before you give an animal a threat, then the animal will confront the stimulus, even if it's not related at all to, it doesn't have to be time locked, excuse me. So you don't have to do it at the exact same time. So this is like getting really pumped up before something, you'll confront it. Or if you stimulate the other pathway rather, in an animal that would normally confront a stimulus, now the animal hides. You can turn them into cowards. So we look at this as kind of coward courage pathway, although it might be adaptive. So it doesn't matter if you stimulate during or before, okay? All right, so speeding through this, you see just as much tail rattling whether or not you stimulate activation of the structure before or you stimulate activation of the structure during the threat. All right? We all know what it's like to be in a good state or a bad state. And I would argue, since this is more in the tenor of this meeting, which is to integrate across areas, that many of the woes of society have to do with people that failed to manage their state well, right? You know, most violent crime is a failure to manage state, right? Rage and things of that sort. Okay, we'll skip the movie for now. Okay, so then we asked, all right, this is all fine, we've identified these brain structures, but what does the mouse actually feel? And of course, you can ask the mouse all day, but it's not going to tell you anything because it speaks mouse, not human. And so we looked and we measured its levels of autonomic arousal. It turns out that its pupil size got bigger, a sign of autonomic arousal. Its heart rate went up and its breathing increased in the condition where it actually was confronting the stimulus. So the highest level of arousal is associated with threat confrontation, which makes sense if you think back to the lion example. You can imagine when you pause and the animal's walking by, your levels of autonomic arousal might be high, but not nearly as high as they are as when the animal's on top of you and you're fighting for your life. So we thought, okay, well, the animals are really stressed and they're fighting. But the crazy thing is, or at least surprising to us, is that if you give the animal a choice of either having this brain area stimulated or not, they choose to have it stimulated. It has positive valence, and it's actually rewarded, we believe, through a connection through the dopamine pathway. So there's something that's highly rewarding about confronting stress in this manner to the animal, such that they prefer to do that than to freeze or to hide, which to us at least was surprising. Those lines just reflect the fact they're spending a lot more time on the laser on side as opposed to laser off. Okay. I'll work through this. We're very interested, I'll work through it quickly rather, whether or not this structure has something to do with phobia and desensitization. You know, we'd love to pair up with clinicians to address that, and we are pairing up with clinicians. I'll talk about how we're doing that in the following slides. I'm going to speed up here. So this is the chamber in which we measure mouse behavior. This is the chamber in which my lab now measures human behavior, because humans can tell you what they feel and think. So Melise Yilmaz, who's here at the meeting with our clinical coordinator, Erin McKenzie, has been putting humans into the equivalent of the mouse box. Sorry, it's a slow motion movie. It was just so you could capture everything. I don't know why programmers don't comb their hair, but you know, someone else can answer that question. So we put humans into this box. We measure a lot of things, and like falling off of a high bridge of this sort in both normal people and people that are terribly afraid of heights or people with generalized anxiety. And we measure a lot of metrics from their body, as well as record from the human brain in partnership with people in neurosurgery like Casey Halpern or my friend Eddie Chang up at UCSF. So this is just, I'm going to move forward here. The other thing is we realized that computer-generated imagery for VR is not great. It's just not realistic enough. And so I had been interfacing a lot with communities, in particular the elite military community, some of whom were in the audience earlier today, although they're elite military, so you didn't know. That was a joke. They were just here to observe the science. And in fact, what you find is that there are all these communities of people that are doing things that are truly scary, and they're doing them by regulating their state in very specific ways. So in interfacing with these military communities and people like my friend Michael Muller, who KJX at White Shark Dive to collect RVR footage, you sort of ask, what are you doing? Are these people really crazy? Next, I think this month, the movie comes out about Alex Honnold, who free soloed, no safety gear whatsoever, up El Cap. If you meet Alex, he's a pretty mellow guy, right? He's not crazy, he's not an adrenaline junkie. So how do guys like Muller do this? I don't really care about his neurology. What I care about are the protocols that people use to put themselves into high stress scenarios that are truly life or death, and that can be exported to people in more typical civilian form that might allow them to maintain calm under conditions of stress. Now, why? I don't have to tell this audience why. It's because you've got high suicide rate at the high school I went to 2 miles from here. You've got tons of stress in the valley. Stress is everywhere and is at the heart of a lot of psychiatric illness and failures of health, let's say, and we need to do something to mitigate it. What you find is that these people are not using brain-machine interface. Some of them meditate, but mainly they're using tools that relate to how they use their visual system and how they regulate their patterns of respiration. So in the last few minutes, I'm going to talk about tools that are out there that my lab is now putting science to, and other labs are putting science to try and understand how to mitigate stress under more conventional scenarios. But what I do understand, and what I've of course experienced myself because I've gone on some of these adventures and done some of this, is that everything's fine and good in life, when you meditate, you're exercising, you're sleeping well, until your pulse rate hits 150. When you're on the freeway, you're in traffic, and you get a text that something might be critically wrong with a family member. Or you're in a long duration stress event, like you have to navigate a cancer diagnosis of a close friend. So how do you do that? How do you regulate and maintain optimal decision making? It turns out elite military is the place to look, because those guys have field tested this stuff, whether or not they knew they were building the protocols or not. They were building them under life and death scenarios. And they also are very interested in evolving those protocols. So this is a recording from the human amygdala done in Eddie Chang's lab of a patient watching this shark VR. So we can get things like breathing, heart rate, pupil size, as well as local field potential recordings from these patients. I'm not going to conclude anything from these data yet, except that we're now in a position to do this kind of thing of measuring from the body and brain of humans in these realistic VR scenarios, which of course the only way you can really bring realistic emotional scenarios to the hospital and laboratory. Okay, so real quick, this is the world of wellness that I've stumbled into, and it's kind of eerie and kind of exciting. You have people that do ice baths every day, and that probably has a lot of interesting effects on metabolism as well as on ability to breathe. This guy's kind of world famous for ice bath stuff. You have the huge success of things like Headspace. I'm definitely interested in mindfulness. The problem with mindfulness is I don't know how to measure it, because the opposite of mindfulness is mindlessness, and I can't really measure that in my lab, or it's hard to do unless you define it as a brain state or rhythm, which people are doing. There's also a lot of people taking drugs now, something I don't condone. The number one question I get as a neuroscientist and interacting with the general public used to be, how do I keep my memory as I age? Now it's, what do you think of psilocybin and ayahuasca? From people 10 years older than I am, 20 years old. My mother is asking me about psilocybin, okay? She's also asking me why I'm not at Rosh Hashanah today, and so that's a different story. But that tells you where this collision, I mean, this is a serious thing, and I have thoughts about this. So the question is, what are behavioral interventions that will allow people to adjust their state, and which ones are available, and what are they, which ones work for everybody, because individual differences are great until they become a barrier to actually doing something, and what sorts of states are we trying to achieve? And here are a number of different arousal states, and I don't have to describe this slide for you to make sense of it, but I think the two things that people really want to know how to do on command is be alert but calm, right? That's nice, and asleep when they want to be asleep. That would be a huge advance for neuroscience and wellness, to be able to achieve those states reasonably well 90% of the time when you want to. Right? Let's forget about emotions for the time being entirely. Okay, I'm out of time, so I'm going to give you a tool now. The tool I'm going to talk about is grounded in the fact that our lab and Mark Krasnow's lab at Stanford and other labs are finding an incredible linkage between the portions of the brain that link emotion centers with arousal centers. So those are reciprocally innovated. So patterns of breathing influence emotional state and arousal state, and the reverse is also true. Okay. Well, let's forget about vision tools for now. So there are three kinds of breathing, and I'm going to just give you these tools. I'm happy to give you the slides and information later. There's tons of breathing: holotropic breathing, Stangraph breathing, apnea-induced breathing, freediver breathing. Here's the take-home message that I think is most relevant here, and forgive me for the people on this side of the room. Breathing protocols that emphasize inhaling are generally going to drive you towards higher arousal states. So if you breathe in for 4 seconds, hold your breath for a moment, and breathe out for 2 seconds, and you keep repeating that, you're going to ramp up your levels of autonomic arousal. If you look at holotropic breathing, we talk about nasal and mouth breathing, closing one nostril, but let's just keep it really simple. The opposite is also true. Exhale emphasized breathing generally decreases arousal. And so you can start to think of it as inhale for a period, that's maybe a 2 count, hold for 2, exhale for 1, hold for 1, and then repeat to increase arousal, lower arousal. Now, what if you want to just maintain optimal, kind of dialed in, calm focus? Well, people from the elite military community call this box breathing. You breathe in for a portion of time, you hold, you exhale for a portion of time, you hold, you repeat. It's a great way to anchor your neurology in sort of an arousal state to stay in the same place and be able to function properly. These ones can be used to drive you up or down the arousal pathway. And there are individual variations. Now, if I could just have 1 minute, I'll give you an important detail, which is how long should you make each phase? I've just put the ratios, 2-2-2-2 or 1-2-2-2. Has to do with your ability to manage carbon dioxide. You don't breathe because you need oxygen, you manage carbon dioxide by breathing. Okay, if I hold your head underwater, at some point your blood starts to feel acid, you want to panic, and you want to take a breath. Okay, so it's all about CO2 tolerance. Who are the best people at managing CO2? Free divers. And so what do? They measure their CO2 tolerance, and you can do this also. So what you do if you want to know your CO2 tolerance, and sorry for the marathon runners, this has nothing to do with fitness, 0. This has to do with how well you manage stress in the moment. So when you wake up in the morning or now, you're going to have very different CO2 tolerance. The way to measure it is you take a couple nice deep breaths through your nose in and out, mouth closed, and then you time yourself how long it takes you to get rid of all your oxygen in a slow nasal exhale. That CO2 blowout time tells you your CO2 tolerance in the moment. It tells you how well you're managing CO2. If you're a skilled free diver, you can go down for minutes. Houdini used to go into a box for 9 minutes doing this, managing his carbon dioxide. He wasn't superhuman. Okay? That's how David Blaine does it too. There's nothing mysterious or magical about it. So if you blow out in less than 25 seconds, I recommend, if you blow out in 30 to 60 seconds, which is how most people blow out in kind of a pseudo-calm, pseudo-alert state, then I recommend breathe in for 5, hold for 5, exhale for 5, hold for 5, repeat. Maybe 20 cycles, 10 cycles, whatever you have available to you, even in real time. And then you can adjust that up. You could halve that or double it depending on whether or not your blowout time is less than 25 seconds or more than 60 seconds. Even though I'm pretty happy and up here and doing this, I can tell you right now, if I were to measure my CO2 tolerance, my CO2 blowout time right now, it's going to be significantly below 20 seconds because I'm doing nothing but exhaling as I'm talking, inhaling, exhaling. I'm in a state of high autonomic arousal. And if you're sleepy, you can run this through the arousal protocol and so forth. So anyway, I've tried to hand off something you can use in real time that's grounded in what we're starting to understand about respiration and arousal. And what I'd like to do, and my hope for the future with this meeting, is we've seen some wonderful talks and important points made about where the gaps are. I'd love to see an intelligent, rigorous bridge, and I think this meeting really represents the first effort at that, between the clinical and the sort of wellness and the things that people generally ask about and care about, psychiatric and neuroscience, psychological, and so, sort of mindful communities, if I dare say it. But anyway, thanks for your time. Sorry to go over. Yeah.